Okay, so I was actually born in a desert in Iran, and then I was living in many different countries before actually becoming a professor in Vienna, where I was living quite a long time at the University of Vienna, and then moving here to France, working for INRA a number of years. And 10 years ago, I started a lab in Saudi Arabia at KAUST. And the reason is really that I was fascinated about deserts and the plants that can survive under these extreme conditions. And I thought that one could actually learn from these plants how their strategies are to cope with heat, drought, and salt stress. This was like the way I set out. And so I'm going to give you kind of an overview of the journey I did over these last 10 years, a scientific journey. And then I think I'll start here, because I think one of the concepts that I realized is that if you are a plant scientist, you can not only look at the plant itself. The plant, obviously, we know is anchored in the soil, but for the last century, people actually thought that the soil is a dead matter, which has, I think, has been completely changed in its perception. So the soil is actually the richest biome on this planet. It harbors what many people know now is harbors a microbiome, which are these bacteria and fungi, but in fact there is a lot more organisms that live in the soil that most people are still completely ignoring in our field. So what is really important is that plants are feeding the entire biome of the soil. So if you take away a plant, there's basically a nutrient depletion in the system. And a lot of the organisms that are normally fed are, of course, dying. So if you want to understand how deserts function, you have to really see that the lack of vegetation has a massive influence on this. Now, many people thought and still think that deserts are something completely natural. But in fact, this is only partially true. And unfortunately, humans have a huge impact on desertification, on the generation of deserts. So deserts are growing all the time. And I think the simplest understandable key is actually overgrazing. So too many animals are brought on fragile ecosystems, and they are gradually taking away the vegetation. And then you have at one point no vegetation, and the whole system actually collapses. So the question is, can one restore regions that are deserts? And we have now 25% of all terrestrial areas is actually desert land, more or less desert land. You will see that desert is a big word, but actually there's many different ecosystems behind that are not exactly the same. So I will take you a little journey through the work that we've been doing. So one of the biggest journey was actually to go into different deserts, collect plants, and then collect from these plants mainly the root endophytes. And I will probably explain you why I'm looking for root endophytes, because I think there's a strategic application potential in using root endophytes and not soil microbes. And then we're also quite interested in working on mangroves, which are very interesting systems because they live between land and sea. And this is, they have one very interesting property. They have their feet completely submerged in water, which basically most plants don't like at all. And we have this flooding problem. And I think here we can see some applications from that side. And on top of that, they are living in seawater, which is like, 800 millimolar sodium chloride. So that's a huge burden. And one of the other projects that we are working on is actually to characterize ecosystems. So over this time, I became more and more also interested in ecology. And finally, also not only in the ecology, but also in the soil. when we realized that actually soil in these desert regions is completely different from the soil that we know here in agricultural regions. So I'll take you onto this journey and start off with the last point, with this kind of what we call the soil bioatlas. And what we did is we got funding to do a representative map of Saudi Arabia. And I should say Saudi Arabia is a huge country. It's about almost 5 times as big as France. So it's almost as big as Western Europe. So we took representative 92 sites and started to look at them in a systematic way. We developed a pipeline for doing GPS mapping. So here, for example, you see this is the Neom region where we have a regreening project. Then we go through this pipeline. We go onto the field. We collect samples, bring them back into the lab, and then we are analyzing them for chemical and physical properties. This is like just a little visualization how these samples look like. So obviously the samples are not all just sand, as you may imagine for deserts, but they are really have quite different structures. And then we are analyzing the chemistry of these, and we have quite nice tools in KAUST. So this is really a big advantage to work there because you have all the technology available on site. So we can do ICPOS for all the elements. We can do XRD for the chemical and crystal structures. And then we go through all these elemental analysis. And then we come to the biological analysis. And so most of the soil analysis that people did was actually not looking into the biology of the soil. And this is actually was one of the things that really was amazing, because we used the classical 16S and ITS sampling analysis where you look at bacteria and fungi, but we also developed a couple of other tools that we can look not only at this, but we can also look at protists, we can look at plants, and we can look at animals that are in these samples. And we realized that these samples sometimes are basically from barren land. So there's no vegetation anymore, there's no animals anymore there. But we can actually generate from just these data a landscape, how the place looked like several hundred years ago. And so we can tell what animals lived there, what plant species were growing there. And this is actually very important for a regreening strategy, to tell the government, okay, guys, and we were very successful to tell them, don't come in with any kind of plant species, but use the plant species that we have identified, which still exist. So we could actually convince them that they are starting to grow in industrial scale plants that have never been propagated. And we are trying to reinstall these plants in these regions. So rhizobia and mycorrhiza are two really important players in this game that we also try to isolate, and by the trapping method, where we give them basically a plant that they can attach to. So we feed them in a way, and then we are isolating them. And so overall, over these many years, we basically now have a collection of about 10,000 different desert isolates. They're all culturable. So we have them all sitting in our lab, in our freezer, and we can use them. And we started actually then saying, what are their properties? And so we started a genomic sequencing project. And to look actually, are they all the same? Because of course, if you look at proteobacteria, you find proteobacteria everywhere, but strains are different. Each strain has some genes that are very specific. And so now we are doing, we started then phenotyping and then doing comparative genomics. And so, when we started this, then we came with another project, which is this native genome project, which is done with a couple of other colleagues, Rod for doing the plant genomics. We're doing the microbiome. Metabolome is done by Salim, plant architecture by Ikram, and I'm also responsible for the soil microbiome. And so here I just brought you a little clip so that you can see what the place actually looks like and what this project is all about, this Native Genome Project. It's really like going through the different landscapes and different ecosystems of this country, which are really variable. And we have overall about more than 2,000 indigenous plant species. So we started actually looking at 50 of them, 50 ecosystems, and we have been working out the basically a map, a biological map of these 50 different ecosystems in different regions of the country. Here you will see a few of these places. So it's not all just sand dunes, as you might think. Really, there we have a big mountain range that goes from north to south over 2,500 kilometers. That goes from low altitude to high altitude, up to 3,500 meters. So you have very big, the biggest diversity, of course, in these mountain areas. And so this, I think, will be a kind of reference system for what kind of plants and what kind of organisms you can find in these systems. All of that will be publicly available. So it's a reference system for the future, because one important thing is also that many of these ecosystems are fragile and they can be lost over time. And given climate change and industrialization, we are really scared that this can happen. So here you just see a few of these plants that we have been looking at in these ecosystems. So it's very different types of plants. We have trees, we have shrubs, we have annuals, and we have perennials. And I cannot go into all the details because that would take you a week to go through the strategies of how these plants actually live. But what I want to show is basically that from these data and from the genomic analysis of all these microbial data, we generated a kind of strategy that we are offering as a consultation for government, which is, okay, if you want to do some re-greening of a place to turn it back from a desert into a ecosystem, into a stable ecosystem, you should start off with right plants, but you should also look into the soil, what is actually missing in the soil. So here we can do a soil analysis and we can actually say what kind of functions are missing in the soil. So we're not only talking about what kind of microbes are missing, but what functions are missing in the soil. And from the knowledge that we have from these microbes, we can basically say, okay, we need this and this and this function. So which microbes can actually provide these functions, right? So some very simple thing is, for example, that we realized there's no nitrogen in the soil. Okay, so that explains on the one hand immediately why 50% of the plants that you find in the desert are all legumes, because they are nitrogen fixers. But it also means that, okay, if we want to regenerate the land, we have to bring in nitrogen fixers, or and the second thing is, for example, phosphate. pH in these regions is between 8 and 10. Everybody knows about physiology that under these conditions, phosphate is completely unavailable. So you need phosphate mobilizing microbes. So this is our strategy that we run. Now, one other question, and that was actually the original question that I had, is can we use these microbes to make crops fit for climate change? So can we do enhanced heat and drought resilience of crops? And so we started a program to actually go right into the field and use one microbe, one root endophyte per different crops. And we used here the station in Dubai that maybe some of you know. It's a very sandy soil, and we tried it on wheat, barley, and alfalfa. And this actually worked quite well, surprisingly well. And we then went back to the lab and looked whether this heat and drought tolerance can actually be also done in a more controlled way. So we went back and we found, yes, that actually works also. Here you see the data from the field trials. And so I think one of the key things is that we can have really enhanced not only biomass, seen here on the left, but really increased harvest. So we could push up the harvest, the yield by 50%. I mean, of course, remember this is desert agriculture. So the yields are very low compared to here. But I think it's very important for these places that we can do that. But it also works very nicely in the lab. So here, for example, for this one microbial strain, Enterobacter strain, that can also, just like here show for wheat seedlings, enhance the temperature priming, the temperature resistance. And then we went from this, because I'm a geneticist and biochemist, I want to know what's the mechanism. So this is difficult to study in these crops. So we go back into to Arabidopsis and really do hardcore molecular biology, if you like. And we take, of course, all the knowledge that is already available from in these systems. And so, for example, for heat tolerance, we know that there are some key genes, some key transcription factors that are regulating heat stress tolerance by directing histone methylation to certain genes. And so we said, is that mechanism actually the same that is directed by these microbes, by this microbial interaction? And surprisingly, it is really. So here, of course, this process is actually happening when you are doing a pre-treatment of your plants with a lower temperature. It induces this kind of memory on these particular genes. These genes then are kind of primed for enhanced expression when second heat stress comes in days or weeks later. And so what we could then realize is that this epigenetic mechanism that is running here, that is induced by these microbes, is happening through metabolic reprogramming of these microbes by information from the plant. So the plant is telling the microbe, do certain things for us. And this metabolic reprogramming produces a little metabolite. And this little metabolite, keto-mesothyl butyric acid, or KIMBA, that is a total natural metabolite, then is converted into ethylene in plants. And ethylene is actually the guy that is doing the epigenetic priming. So if you knock out ethylene signaling, we lose this beneficial effect. And by the way, we also discovered that ethylene mutants are really hypersensitive to heat stress, which I think you know. So you can also discover very basic things by working with these microbes. Yeah, so this was the discovery for heat stress. And here I just show you something for water. So we did water stress here, for example, shown for alfalfa field trials, and then bringing that back into the lab and looking what's happening if that also works on Arabidopsis. And you can see when we do these treatment of, so here you mimic drought stress by putting the plants on 25% PEG, standard procedure. These plants really don't like that. They really are stopping to grow. But if they are colonized with this root endophyte, they are having a massive growth. So what is happening here? We were really stunned about this because we found that relative water content was much better. They still have a considerable transpiration. They do not close stomates as what you see in drought-treated plants. And the most important thing is we found that water use efficiency was pushed up. So these plants are just able, the Arabidopsis is just able to actually get water out of the dried soil to a much higher extent than the plant that is not colonized. So what's behind that? We did, of course, a genetic analysis and found that this is epigenetic priming again. So these plants are doing something that you don't find when you colonize them. There's absolutely no change in gene expression unless you come with a stress, which is in this case drought stress. Then you have massive changes in gene expression that are all dependent on ABA. And so here this metabolic reprogramming happens to produce we have a number of carotenoids that are produced that are probably modifying this ABA pathway. So if we knock out the ABA pathway, we lose this epigenetic primer. And so second evidence that we have epigenetic mechanism that is driven by a microbe. Third example is maybe the most crazy thing that I ever did, is to take these mangrove microbes and then put them onto rice. We said these plants, rice, are also underwater, at least to some extent. And but we have also used a rice variety that is very sensitive to salt, and some rice varieties are sensitive to salt. And if you just look here, what's happening when you have hydroponic or soil conditions, you see that these plants are, so these are two different microbial strains that we isolated from the roots of the mangroves. And if we use them, we get actually a number of grains, the weight of grains improves, and you can see that here nicely. This is under normal non-saline conditions. But one thing that was really interesting is that we saw that there's also an effect on the life cycle of these plants. And if you incubate them with one or the other, or the combination of the two, you can actually reduce the life cycle by 1/3. That means from roughly 90 days to 60 days or something like that, if I see that right. So that would actually mean, but at the same time, you're having a better harvest, better performance of these plants. So it's quite amazing. But in terms of application, if you think about that, 3.5 billion people depend on rice. In tropical systems, they could actually have a third harvest. And that would already be the solution for all our 50% increase of producing more food. What happens if we put the plants onto salt stress? Now, these plants don't like that at all. And if we have the incubated with them or colonized with these microbes, we see a massive increase in the salt tolerance. You can see here the weight of grains is about threefold, number of grains also, and you can also see the quality. So, and this paper is under revision, and I just want to say that the mechanism behind is epigenetic. Isn't that fascinating? 3 different systems, completely different microbes, different physiological things, all epigenetics. I think that's fantastic because it's such good news in terms of the trade-off. The trade-off that we often have the problem with is that we are pushing up resistance for abiotic stress or biotic stress resistance, but we are losing the growth and the yield. And in this case, this is not the case. Why not? Because the genes are actually only coming in when they are needed. The epigenetics is the key thing, that you target genes, but you don't express them unless you actually need them. The point that I still want to make is orphan crops. So I think we have to look more into orphan crops. There's a lot of orphan crops in the world that is not in our world because it's in the less developed countries. These are mostly subsistence farmers that are using this, have been using that for hundreds of years or thousands of years. And so it will just show you a study that we just published recently. So it's actually a millet that is grown in West Africa. And this is a collaboration with my colleague Simon Kratinger, who is a fantastic young geneticist working at cows. And he got hold of 123 different accessions that are all growing here over a vast region in Western Africa. So this is stable crop for these people. And but if you look, very interestingly, if you look at the yield, and over time, has not been this. So this is a not domesticated crop. There's no improvement whereby wheat, rice, maize, they all are improved over time, and of course you have better harvest, but not in this case. Why? What's wrong with this? So first of all, there is well, we have some advantages because this can grow under nutrient poor soil and is very drought tolerant. But if you look at the one of the grain size, it's tiny. So one of the key things one could think in terms of domestication is push up grain size, right? So here we were doing two things. We were actually looking whether the domestication, so that's Simon's project, but we were looking very much into the microbial community. And I'm telling you in one moment why this can be so nicely. So this population that we have genetically, so it's all sequenced, completely sequenced, and you can see that the genetic structure is very interesting because it really follows the different countries. And it follows not only the different countries, but it really has a very nice correlation with the water, with temperature, with soil structure, with latitude. So there is very good data set for that. So that we thought this is a fantastic set of material to actually ask this question. Does the seed microbiome co-evolve with plants to help in the adaptation to environmental conditions? So here we have a set of seeds that has one advantage. It really comes directly from West Africa, and it has not been touched by, breeding. And so we have basically now the possibility to look into these local adaptations from not only from the plant genetic side, but also from the microbial side. And so we took the most diverse representative 18 different strains, varieties, and we were looking into the seed microbiome. If you don't know about seed microbiome, every plant has a seed microbiome. But so far, it is considered that most plants have different seed microbiomes. And we basically don't know nothing about their functions. So here the question is, do these seed microbiomes convey different functions? The first answer is yes, you can easily see they have different variety of different microbes. So these are different, just look at the colors and you will see that they have very different microbial diversity. And then we looked whether the genetic structure of the plant actually is correlated with the microbial structure. So this is basically maybe a little bit complicated, but what we found is that we have a core microbiome. So that means all varieties, irrespectively where they come from, have the same 191 ASVs or strains of microbes. But then if you look into the other genetics, they have quite different structures. Now, what does it mean? And what happens actually to the seed microbiome? Is the seed microbiome in a particular variety fixed and stable forever? Or is that dynamically evolving with the conditions of the plant? So here we did an experiment where we used now 3 different varieties with 3 different seed microbiomes, and we propagated them on a rich soil, right? And then we looked into the next generation of the seed microbiomes. And we asked the question, does the seed microbiome stay the same, or is it changing? And the answer is, some of the guys are clearly staying the same, but some guys are changing. So that means basically the seed microbiome that a plant has available as an accessory genome is in a dynamic way associated and can be exchanged also. Interestingly, some of the guys are lost, and some of the new guys from the soil are actually coming up and ending up in the seed again. So it's quite interesting. So next question, do the seed microbiomes that we have identified here, do they actually correlate with the environmental conditions of the plant? So does, for example, a plant that grows into soil that is salty, is that having a particular seed microbiome? And so we looked into these different parameters, and you can immediately see that there are different colors for different, so we found clearly a link to the soil quality. That means if you have a soil that is more solid, more compressed, you will have a different seed microbiome. If you have a higher temperature where your plants are growing, you have a different seed microbiome. So that means these plants are using these seed microbiomes as an accessory help for, well, adapting to local conditions. Of course, if you have now data available of sequence data available, you can also do GWAS. And so we did GWAS, and we found actually a number of genes that are quite interesting. Because what would you expect for a seed microbiome? Well, we know that immunity is a very important player, right? And so what we see here is that we found a number of important immunity guys. For example, we found WAC2, well-associated receptor kinase. We found RBOHC. So if you work in immunity, you know exactly that these guys are very important. But we also found a number of genes that are actually more involved in development of the seed or of the embryo. So this really sparked our interest, and we could not really go further with this project because there's a orphan seed and very difficult to transform and to do crosses. So what we did is now we went to Arabidopsis, and but I'm running out of time, so I'm not showing you these data. We have to wait for that. But I just want to say this is my take-home message, which is basically that if you want to look into plants, you have to look into more than that. You have to look not only into plant genomics, you also have to look into the microbial genomics that is associated with a plant. So that means if you want to work on a plant, look into the seed. Here you have tons of information. But of course, if you grow your plants, you also have to look into the microbes that are associated, that are in this dynamic system. So obviously, soil, geographic localization, climate, everything has to be somehow integrated into this concept, which unfortunately makes life a lot more complicated for you guys, but I think it makes it also more exciting. So with that, I think, well, I just want to thank the people that have been working with me, the collaborators on the bottom, but also my group of people here. So this lady was doing the last microbiome work here that we recently published, and the bioatlas was done with this guy and here Maged, and yeah, so we're really a big team of people, and everybody is happily working together from different countries. Thank you.